Hello. Let's continue with our discussion on oncogenes and cancer. We have 7 types of proteins controlling cell growth and proliferation. This picture shows these stages, the proteins that is interacting at various stages in the cell. The first one is extracellular signaling molecules, maybe growth hormones or some other proteins that will act as signals for the gene expression or regulating the gene expression in the cells. So they are signaling molecules, the first one. Then signal receptors. Of course, if there are signaling molecules, there should be signal receptors which will receive the signals and transfer them to the cell. So second stage is the signal receptors. Then signal transduction proteins. Signal transduction proteins or intracellular transducers are the proteins that carry the signal from the signal receptors to second messengers and subsequently into the nucleus, where it will interact with the DNA and subsequently with the other transcription factors and other things. So these third category is signal transduction proteins or intracellular transducers. The fourth category is transcription factors. And during the transcription, we discussed about the transcription factors which will interact with the regulation of gene expression in eukaryotic systems. Then there is apoptotic proteins, which may include tumor suppressors which promote apoptosis, and oncoproteins which promote cell survival. So both were apoptotic proteins. Of course, it is having there should be a balance of apoptosis and cell survival, for which it requires tumor suppressor proteins and also oncoproteins. Then the next stage is cell cycle control protein. So of course, here is transcription factors which will act on the level of transcription, and the next stage is a cell cycle control proteins, which will decide whether the cell cycle needs to go for a cell cycle arrest, or whether some repair mechanisms need to be activated before going to DNA replication and cell division. So all these things were actually decided by the cell cycle control proteins. Then DNA repair proteins. So if there is some errors in the DNA replication process, then DNA repair proteins will repair that error, and subsequently that error will be rectified. And finally, we have the mRNA that is produced, which will be translated into proteins, and that proteins also include apoptotic proteins, which will result in programmed cell death. So the virus encoded proteins, usually if it is a viral DNA, that will be encoded into proteins, and viral encoded proteins, usually they activate cell receptors and induce cancer. But normally these are apoptotic proteins or other cellular proteins which will have normal functions in the cells. So here you can see that the control of cell growth may be by influencing various stages, starting from the cell signaling to finally till the production of apoptotic proteins. Genes commonly mutated in cancer, as we discussed, that may include 7 categories or 7 or 8 categories. The first one is, as we discussed, that is gain of function mutations that turn proto-oncogenes into oncogenes. So the gain of function mutations may affect wherever. What are the stages? You can see that growth promoting signaling molecules. So signaling molecules can be affected by uh, gain of function mutations. Receptors, the second stage, that can be affected by gain of function mutation. Intracellular signal transduction transducers can be affected, or transcription factors associated with cancers. So all these four stages can be affected by gain of function mutations. Then loss of function mutations in tumor suppressor genes such as cell cycle control proteins. They are tumor suppressors. So cell cycle control proteins, DNA repair proteins 5 and 6. These are cell cycle control and DNA repair proteins. Or example, DNA repair proteins are also known as caretaker genes. And the next one, or anti-proliferative factor receptors such as TGF beta, etc. receptors can cause cancer. So these are different tumor suppressor genes, and the loss of function mutation may affect this type of proteins. Then loss of function mutations in pro-apoptotic proteins are associated with the cancer. That is also available. That's affecting the stage 7. Then gain of function mutations in anti-apoptotic proteins is also another stage in which such a mutation may occur in cancer-related genes. Although relatively rare, the human cancers can be caused by retroviruses that carry dominant viral oncogenes. And more commonly, the insertion of a strong retroviral promoter upstream of the promoter oncogene can switch it on and leading to cancer. So the viral oncogenes also having a significant role in the contribution to cancer, and their insertion, these viral oncogenes, into a strong retroviral promoters. These proto-oncogenes, which is actually taken from the cellular oncogenes, may be the viral oncogene. And if it is placed under the control of a strong retroviral promoter upstream of the promoter oncogene, then it may lead to cancer. So the cellular oncogene now carried over to the virus, which is an oncogene, viral oncogene, and if it is placed under the control of a strong retroviral promoter in the virus, then that combination will become a strong cause for cancer. Oncogenes and mutated or deleted tumor suppressor genes disrupt signal transduction pathways to induce cancer. So that's another chance where such mutated or deleted tumor suppressor genes may disrupt the signal transduction pathway and induce cancer. In spite of a diversity and complexity of the signaling pathways, they have some common features. Growth factors bind to receptors and activate them. Activated receptors trigger a series of protein phosphorylations and other complex chain of events. Protein phosphorylations trigger changes in transcription factors that alter the expressions of specific genes. Activated or inhibited genes produce proteins that influence cell proliferation and cell death. So this is an illustration which shows the interaction of various factors such as regulatory elements in the cell. Here, for example, you can have the growth factors, anti-growth factors, cell death or death factors, cytokines. So these are external stimulus or hormones, survival factors. These are different external proteins that is interacting with the cell to activate the cell growth or even regulate the cell growth. So like growth factors, anti-growth factors, or cell or death factors, or apoptotic factors, and all these are different factors which is interacting with the cell surface receptors, so that a proper signal for cellular growth can be conveyed into the cell. So that cell which receives these through proper receptors, cell surface receptors, will transfer it through a series of cellular transducers or some factors that are messengers, second messengers, which will carry this message into the nucleus through various pathways. So we have various transducers or second messengers which is available that will transfer this into the nucleus, where it will interact with the DNA. And finally, when it interacts with the DNA or the transcription factors, that changes its gene expression. And that's the regulation of the gene expression by the transcription factors, which will be activated by the signals which is received through these pathways. Now the gene is expressed, and that results in cell proliferation and production of the various proteins. Then if there is a damage, then damage will be repaired, damage sensor will be there, damage will be repaired, and all these things will occur in the nucleus. Now there is cell death or apoptosis. When if it is a normal cell, the apoptosis will be there, the cell death signals will be received, and cell will be leading to cell death or programmed cell death or apoptosis. So this is the overall picture of the interaction with the different factors or growth factors or stimulus which is received by the cell surface receptors and subsequently transferred into the nucleus through the cellular transducers and second messengers, and finally the cell or cellular expression or gene expression will be regulated in a coordinated fashion. So this is another illustration which also works in the same way, and this is about the MAP kinase, and you can see the array of different factors that is involved or interacting with each other in regulating this gene expression. Here, you can take the example of MAPK or mitogen activated protein kinase and ERK pathway, and you can see the various stages. The first one is a MAPK kinase pathway chain of events in 6 steps for a signal communication from a receptor on the surface of the cell to DNA in the nucleus. So that pathway is illustrated well here. The first one is a growth factor binds to the receptor. So this is external growth factor and external growth factor receptor, which is present in the cell surface membrane. So this growth factor receptor will receive the external hormone receptor, so hormone or some growth factor, and that will be received and that will be conveyed to the cell. And then clustering and autophosphorylation of the receptor will occur. So clustering and phosphorylation of the receptor will occur. Then activation of the RAT sarcoma or RAS protein. So this system will activate the RAS protein. So once the RAS protein is activated, the RAS protein from its inactive GDP bound form will be converted to GTP bound active RAS protein. Now activation of the cascade of protein kinases will be affected by, as the effector molecules will be activated by the GTP bound active RAS, and that includes MAPK kinase, that's mitogen activated protein kinase, and other set of different kinases, RSK, MNK, MEK. These are different kinases which will be activated in the cascade of events. Now activation of the nuclear transcription factors finally. So finally the different factors and transcription factors will be activated, and that will be transported through the to the cytoplasm, to the nucleus. So once the transcription factors were created and are activated, and it is passed into the nucleus where it will interact with the DNA and regulate the transcription process. So that includes the synthesis of cyclin and CDK molecules. All these things will interact with the DNA and regulate the transcription of the gene. So this is about a point mutation that that is seen in case of a RAS oncogene. In the case of a normal RAS oncogene, the DNA from the DNA mRNA is produced, and RAS protein, when it is produced, here is a glycine. Whereas in a oncogene, this is a proto-oncogene where glycine is used. In the oncogene of the RAS, there is a mutation which results in replacement of the glycine with a valine. So this is a mutant of the RAS gene, which will have a constitutively expressed state, which doesn't require the GTP GDP activation. For example, as we discussed in the previous one of the slides. Okay, in this slide, as we discussed here, the RAS, GDP bound RAS, inactive RAS is activated by the signal which is received by this one. So if the signal is not there, this phosphorylation and subsequent conversion of these inactive RAS to active RAS will not be occurring in normal system. But in such a case where a mutant RAS gene is there, it creates a constitutively active RAS protein which doesn't require the external stimulus and subsequent phosphorylation. So, mutant RAS gene is a constitutively active, continuously passing its message to the effector molecules, so that the different kinases will be always expressed and gene will be in a continuously active state. So this is about chromosomal translocation that creates a Philadelphia chromosome we discussed earlier, from where the BCR gene from the chromosome 22 is placed nearby the ABL gene in the chromosome 9, and this exchange or this translocation results in a Philadelphia chromosome. So after the translocation event, now we have the Philadelphia chromosome in which both the genes were placed together. So which results in the production of a fusion protein, and we have a large amount of fusion protein that is produced here. So here it is another example in which TRK oncogene is created by the chromosomal inversion event. So here we have a NGRK1 that codes for the receptor with tyrosine kinase activity. So this is a TPM3 which codes for tropomyosin. So this is a tropomyosin gene, TPM3 gene, which codes for tropomyosin, and which causes the receptor region to form a dimer. So this is a tropomyosin gene. This is the kinase gene, or tyrosine kinase gene, the second one. So when the DNA breaks here and that sequence undergoes inversion, so now the entire region is inverted, resulting in joining of these two segments. So now we have here we have a TRK oncogene. TRK oncogene is actually a portion of the TPM and tropomyosin and another portion of a tyrosine kinase. So a fusion protein of a tropomyosin and tyrosine kinase will be produced from these segment. So that is represented here. You can see that from the coil-to-coil region of that is produced from tropomyosin, the initial segment, and the segment part which is contributed by the tyrosine kinase will have a tyrosine kinase receptor, which will be always in a phosphorylated state. Phosphorylated form means that is a constitutively active form. Here the same thing is represented. Normally such receptors, actually it requires a growth factor when it is present on the cell surface. This is a receptor, tyrosine kinase receptor, and that receptor requires a growth factor. When the growth factor is there, then it changes its conformation and it changes to the phosphorylated active receptor. But a mutant will lack this provision, so it will be in a constitutively active state with the phosphorylation and the fusion protein. The fusion protein, this region is contributed by the tropomyosin, and this is by the tyrosine kinase. So the gene is in a constitutively active state, and this doesn't require any external growth factor stimulus. So this type of mutant receptors is the reason for a constitutively expressed state or mutant state. So the second one is amplified receptor. So in this case, the number of receptor molecules were increased, or multiple copies of the receptors were created, and it also increases the overall gene expression rate. So this is about proto-oncogene receptors, for example, the HER2 receptor and EGF receptor. Two receptors were represented there. Usually, in the normal state, there will be valine represented here, but a change or a mutation from valine to glutamine, valine to glutamine result in an oncogenic mutation. So, an oncogenic mutation, that's a change of a valine to glutamine in HER2 receptor that results in the production of a constitutively active state. So usually it requires some external stimulus, but it is no more required once this change of a valine to glutamine is done. Here EGF receptor is there, and in that case here is an inactive receptor without any external stimulus. It is not active, but a deletion event of the receptor component, external receptor component, and that render the entire molecule in a constitutively active tyrosine kinase activity. And this is another example. We already discussed this one, the RAS kinase, in which here we have the RAS protein. Here is the inactive state of the RAS with the GDP that is converted into active state by GTP binding. GEF is the factor that is supporting, and usually there is a GAP proteins that facilitates the degradation of these GTP into GTP bound state to inactive GDP bound state. This is a GAP protein family. But if there is a mutation that results in this blocking this conversion or this GTPase activity, what happens is that it will be stuck, or these RAS proteins will stuck at the active state for a long period. So that means now it's in a cancer state or in an oncogenic state in which the RAS protein family proteins is always in an active state. So here is some viral activators that is affecting on growth receptors. For example, here we have erythropoietin receptor. Erythropoietin will be received on the cell surface by the erythropoietin receptor, and subsequently the gene is activated normally. But when instead of the erythropoietin, a viral protein GP55 that binds to this receptor, now this is turned on to the constitutively active state. So this is another representation in which genetic instability of cells using different mechanisms. For example, here the first one is the excision repair of the gene, and the excision repair usually the first mutation result in change of one of the genes. The second mutation, if both the genes or both the alleles were mutated, then that results in that results in the conversion of or disabling of that particular gene into an unstable one. So genetic instability that results in such xenoderma pigmentosum, for example, one disease. And another mismatch repair gene, for example, another one in which the first mutation will change one in the first allele, the second mutation will change the second allele, and that results in the genetic instability, which results in hereditary neopolyopsis or colon cancer, etc. So various genetic instabilities were the result of excision repair genes or mismatch repair genes. So these are defective DNA repair genes. Usually DNA repair mechanism or excision repair mechanism or mismatch repair mechanisms, that genes were supposed to function in the cell so that the corresponding corresponding type of repair is always available, or that machinery is available in the cell. But if any of the genes that is required for excision repair or mismatch repair is disabled, that result in genetic instability, so susceptibility of the cell to further mutations, and when it is further exposed to a carcinogen or a mutagen, the cells will be immediately affected, and that results in various types of cancer stage or disease. The next topic is on tumor suppressor genes that we will discuss in the next session.